Thank you for joining the team at Hands-On Labs today for our discipline-focused live demo series. Today's topic is dedicated to HOL's Anatomy and Physiology online curriculum and kits. Before I hand the mic over to our host, let me take a moment to review the webinar logistics and give a brief introduction on today's event. We encourage our attendees to participate in the conversation by asking questions and sharing resources. Please use the chat function to join in the conversation. Be sure to toggle the window to all panelists and attendees if you'd like the entire group to see your comment or question. If you have questions, add those in the Q&A box. You can find this section in your toolbar. We have several distance learning specialists on the panel today to help answer any questions you have in real time. For questions pertaining to kit cost and customization, please work directly with your dedicated distance learning specialist once the webinar concludes. Dr. Duane will address any remaining questions at the end of the demo so you have clarification on important topics. We'll start the presentation with a quick poll to gauge our audience temperature on their past experience with online teaching. To participate in the polling segment, submit your response when you're prompted with the poll pop-up. At any point in the conversation, you can raise your virtual hand and our distance learning specialist will send you a private chat message to support your needs. All attendees will be entered into a raffle to win a $100 Amazon gift card. We'll share the lucky winner in our follow-up email at the end of the day, along with a recording of this webinar for you to reference moving forward. So, what can you expect to see today? During our A&P live demo, our Chief Resident Scientist, Dr. Duane Cagle, will help you explore the human body and its functions with HOL's rigorous online curriculum and the powerful platform that hosts the content from both an instructor and student perspective. Before I introduce our host and distance learning specialist, let's get an idea of our audience's proficiency with online lab learning by launching the poll now. All right, it looks like we have a nice mix between instructors who have done this in the past and those who are new to distance learning labs. Thanks for participating. If you don't already know our host today, Dr. Duane Cagle is our Chief Resident Scientist and has been the brains behind Hands-On Labs curriculum development for the past 9 years. We also have several HOL distance learning specialists on the call today who will be manning the chat and providing helpful resources throughout the webinar. A warm welcome to Pam Paisawang, Vanessa Clower, Justin Johnson, and our main host, Dr. Duane Cagle.
Thank you, Justin, for the warm welcome. I'm so excited to share with our audience today our A&P curriculum and our course delivery platform, HOL Cloud. So let's get started and take a quick tour of our curriculum and platform as a student would encounter this when using one of our kits. Great, so you should be seeing a landing screen now for a student once they enroll in our online lab manual. So very excited about A&P. This discipline really goes back to our beginnings. And if you've taught A&P online before or currently, or know a little bit of history about online science laboratory education, you may realize that the A&P discipline and other allied health science lab type courses were really on the forefront of online lab instruction. And indeed, that's true for our company, Hands-On Labs. So we have a long history with A&P. Our curriculum has become quite standardized over the years. A&P is one of those disciplines where it seems like most every institution teaches the same topics and concepts throughout a semester. So it's really easy for us to mold our curriculum to fit the needs for the vast majority of our clients. So how does a student use our materials, and how do they use our kits along with our curriculum to successfully complete their online laboratory requirements? So we design all of our curriculum to work in conjunction with the physical items in our kits, and we house all of this on our online cloud-based learning platform called HOL Cloud. And what you're seeing right now is the student landing page, and you'll notice in this sample course that the student has 5 lessons to complete, along with 3 prerequisites. And then on the landing page, they see any notes from their instructor. And these instructor notes appear in real time. So as soon as the instructor saves the note, it appears for the students in their view of the landing page. So here the instructor is just welcoming students to the class, making sure they realize they have to complete their prerequisites before they begin any of their laboratory experiments. We know how you teach this on campus, and we also know that a lot of you have some level of trepidation, perhaps at the thought that you've got to take your campus-based laboratory course online this coming fall, or even late summer for that matter. And we know that safety is really at the forefront of your concerns. Now, not unlike you have a requisite safety lab day, usually the first day in the laboratory for your on-campus students, we have a lab safety prerequisite that students cannot advance to any of the labs for the course until they complete the lab safety lesson and sign the student safety agreement. Now, speaking of safety and knowing of your concerns regarding that, I do want to point out up front that we provide liability coverage for the end users of our lab kits as long as they conduct the procedures as prescribed in the lab manual. And we'll take a look at those procedures here momentarily. Also in the prerequisites, you'll see 2 other titles, one called Getting Started. And this assures that students are familiar with the learning platform technology and how to submit their work, how to navigate between sections, and just all in all, how to become familiar with all the features and functions of the online lab manual platform. We also know that many A&P courses have some element of microscopy at some point during the course, usually in the first semester especially, if it's a 2-semester course. We'll let you know up front that our curriculum is microscope agnostic, meaning that your students can purchase a microscope through our company. They can use a microscope that they already own or have access to, or if you choose as an instructor, all the students in your course can use our interactive virtual microscope in place of a real microscope. And if we have time today, I'm happy to give you a tour of that. But because it is a technology feature, it becomes a prerequisite on our lab manual platform. Now, once a student's completed all the prerequisites, the lessons are unlocked for them. You, the instructor, can choose what order the students conduct the labs in and whether they have to conduct them in the sequence or they can randomly move around. And I'll show you some examples of that in a few minutes. But let's start with the first lesson in this student's account, the cardiovascular system, blood. And as a student, when we click on a title, we get a drop down with 3 sections. One section is duplicated here, but there's 3 sections, and they're labeled exploration, exercise, and evaluation. And indeed, we model our curriculum after the 5 E's of science learning, and when we click on any of these sections, you'll see a further example of that. So 3 sections per lab: exploration, experimentation, and evaluation. Again, modeled after the 5 E's of science learning, we include 3 of those within our laboratory curriculum. Now, before a student does anything, we want them to be aware of how much time they need to allocate to be successful. Time allocation is a huge challenge for online laboratory students. You know, they don't have a pre-described 1 to 3 hours per week block of time that they have to show up on campus and be in the laboratory. It is really upon themselves to properly set aside the proper amount of time throughout the week for them to conduct these experiments in their home or office or wherever they may find themselves. Traditionally, or typically, our exploration sections take about 30 minutes for the student to complete. Our procedures or experimentation section will take anywhere from 1 to 3 hours, depending on its complexities. And then finally, the evaluation or final exam for the lab should take the student again about 30 minutes. Before a student begins even the exploration section, they are presented with very detailed learning objectives so they can conceptualize what they are expected to accomplish for each independent section of a lab. If you have a background in education, you may recognize these learning objectives in how they're formatted and that they conform to Bloom's taxonomy. They begin with a verb. The verbs also are formatted within the larger constructivist learning theory. So in our exploration section, those verbs are a little less active and connotate a lower level of understanding. So terms such as describe and explain and outline. We'll see these verbs become more active-based and demonstrate a higher level of comprehension in the upcoming sections of the lab. Now, before a student begins the exploration section proper, they are presented with a blue button at the bottom of their page, and it's labeled Pre-Test, Test Your Knowledge. This specifically is a group of formative assessments. These formative assessments exist as drag and drop activities for the student, and they can consist of ordering or sequencing that we see here, or we'll see some upcoming examples of matching and categorization. You know, studies show that for an online student, a drag and drop activity is highly engaging. So we want to make sure we engage the students as much as possible with the first activity that they complete per lab, which is a formative assessment. Now, if this was a live student account versus a demo account, I could actually manipulate and move these answers. And then once I completed that activity, the system would give me immediate feedback so I can identify as a learner which selections I answered correctly and, more importantly, which ones I got incorrect. The whole purpose of these formative assessments is not necessarily to grade your student, but to make them aware of any knowledge gaps they have of important concepts that are addressed in the procedures of the lab. And studies show that once a learner can identify knowledge gaps, when they encounter that information in the future, they will automatically focus on it. So here's an example of a categorization question. So categorizing features of either red or white blood cells. And then lastly, here's a traditional matching exercise for terms and definitions. Again, these are always drag and drop in format. And once a student answers these questions, they are always provided with immediate feedback. Here's a labeling example where they're placing the correct term within the table. Now, students will see all of this information in the upcoming exploration content pages. We only test students on content that we provide. And as curriculum designers, we only provide content that is pertinent to the laboratory experience, meaning the upcoming procedures. But rest assured, we know that some students in online courses do work ahead in their laboratories and may attempt to complete a laboratory before the topic is covered in the lecture portion of the course. And because of that, we're going to review all important concepts with enough thoroughness that students should be well off from a comprehension level to be in a position to understand their experimental results and even why they conducted the procedures in the first place. So if your students do need to work ahead, we will set them up for success. So as a student, once I complete those formative assessments and I've been made aware of concepts I don't yet understand, I then encounter the background content for the lab. Again, we title this as exploration. You can think of it as the introduction or, again, the background section of a traditional lab manual chapter. And how we design our curriculum in this section is always in one or at most two paragraphs of text with all defined terms in bold print. And those text paragraphs are always accompanied by visual elements such as diagrams that we see here, or graphs, equations, photographs, but just some type of graphic representation of the content that's provided in the text. And studies again show that anytime a student can visually relate to text, their comprehension levels are increased. So we always want to give students visual references. Now at the bottom of every content page, students are going to encounter another activity. And this activity button is labeled question time. And the question time button includes one or more multiple choice questions. And these questions are formatted around content that is provided on that page of the exploration. So the goal here is to test students' understanding of key concepts as they are presented within the exploration section. Again, if this was a live instance, I could click on one of these for choices, and as soon as I hit submit, the system would alert me to the results of my efforts, meaning it would let me know that either congratulations, I got the question correct, or more importantly, if I missed it, what the correct answer should have been. Now, you can choose to grade these as an instructor, and you can choose the point value you assign to them. And I will show you an example of that momentarily. So there are a number of background exploration content pages in each lab manual chapter. This particular one, 5, has 6 pages and then a review page. I'm not, for time's sake, going to walk through every one of these content pages, other than to click on one or two to show you that indeed they are all formatted in the same fashion, and that they're manageable blocks of text with all defined terms in bold, always accompanied by a visual element. Now, the final page of the exploration section is a review for the student. It is a summary of all the questions that they answered. And again, these will be marked as correct or incorrect. And if they are incorrect, the correct answer will also be marked so that students have one last opportunity to review all the key concepts before beginning the experimental procedures. And those procedures reside in the experimentation section of the lab manual. Again, when a student clicks on this section, they are provided with the estimated completion time. Very, very important. Students can't jump around, by the way, between each of these 3 lab manual sections. They always have to complete them in sequence. So as a student, I can't simply click on experimentation and start setting up my lab procedures unless I have successfully completed the exploration section. Likewise, I cannot take the final exam until I've completed the prior 2 sections, exploration and experimentation. As promised, we have new learning objectives now as a student for this section, so we can conceptualize what we're expected to accomplish by its completion. And the verbiage here, again, in accordance with Bloom's taxonomy, shows a more advanced level and more active level of comprehension. Now, before a student begins any procedures, they're provided with a reminder of all the materials they need to have set aside. Not only materials in the kit that we provide, but also common household items that the student should already have on hand. We do not provide things like distilled water, dish soap, or again, common household items like paper towels or regular paper. Those are expected of the student. The materials that we provide as a science company are just that, science materials. So things such as microscope slides, staining trays, staining depression slides for blood typing, which is this experiment is an example of, and then a variety of chemicals and reagents and other labware that are unique to the particular experimental procedures that the student is about to conduct. I do want to point out, and we will see some examples of these materials in the upcoming pages, that whenever possible, we package experimental chemicals in unique bags clearly labeled for the experiment they're to be used in. Your students are not receiving a bottle of chemicals that they have to then pour into smaller containers and use throughout the teaching term. Everything is individually packaged. This allows students to only work with small volumes of reagents, which is much safer, and also greatly reduces the chance of spillage and waste disposal at the conclusion of an experiment. So if we give the student 3 mils, for example, of the right stain and buffer that you see at the bottom of this table, they're using approximately 1.5 mils per procedure. They can repeat the procedure if they need to, and therefore, if they're disposing of anything from a chemical standpoint at the end of the procedures, it's less than 1 milliliter. So very, very safe. We're not creating disposal issues for the student or the jurisdiction that they reside in. So the students now set aside all their items. They are ready to begin the procedures. And this particular lab consists of 2 exercises. The first one involves microscopy, and the second one involves blood typing. Now, throughout our procedures, whenever possible, we do include videos of standardized lab procedures. So, for example, if we have an experiment that requires microscopy, it is going to coincide, and I'll mute this, it's going to coincide with our microscopy video to assure that in this instance, for students using a real microscope, that they get a quick refresher on basic operating principles of a microscope. Once a student has viewed any of the requisite videos, they are ready to begin the procedures. The procedures are written in a very straightforward, purposeful manner, because we know that the #1 challenge of your remote science laboratory students is they cannot get an immediate answer to a question they may have. Unlike their on-campus peers who can simply raise their hand and have their instructor or TA walk over and explain something to them, that is not an option for remote science students. So it is imperative that we include very detailed procedures written in a very straightforward manner so that they're easy to understand, and each step is a call to action for the student. Any explanations that are needed beyond that are always included in bold text, and they're labeled as a note. And then throughout the procedures, we do provide photographs of individuals conducting the same procedures, so the students are visually guided through the step-by-step instructions. Again, the goal here is to reduce student questions and therefore prevent student frustration. We want your students to be focused on the experimental outcomes and the data that is generated and not lose focus because they're frustrated and have questions that they can't find answers to. Now, throughout the procedures, your students are going to encounter references to photo panels or data tables that you see here, always in bold print. Now, these photo panels and data tables are always going to be located in the lower right-hand corner. This is important for two reasons. One, it prevents a student from having to keep track of individual documents in order to record their work and then turn around and submit to their instructor. So no creating a Word document or, you know, any type of file like that to submit their work on. It's all integrated into the cloud-based lab manual. Another important reason for having this expandable data tab at the bottom right of the screen is for students who are not using a traditional computer with keyboard. For those students that are using mobility devices such as tablets, it's very easy for them to then tap the data button and they get a nice full view of the data table or an image panel that is then easy for them to work with. And indeed, we have made our lab manual mobile device compatible. So whether your student is using an iPad or an Android-based tablet, they have as easy access to the content as students using more traditional desktop or laptop computers. Throughout our procedures, students are expected to not only complete data tables of their experimental results, but also upload photographs of their completed work. You know, I get a lot of questions over the years from institutions and instructors looking to take their classes online, and they always have a very real concern, and that is, how do they assure that students are actually conducting the experiments? Well, you know, best practices is to always require students to upload visual proof of their procedures and of their results. And whenever possible in our curriculum, we require that of students. And we'll see examples of students' uploaded work in just a moment when we go into the instructor and grading view for a particular exercise. Once a student inputs their work, and by the way, any student input is auto-saved within these data tabs. So every 3 seconds, it saves the student's work. The student never has to hit save. The student does not need to worry if they lose internet connectivity for any reason that they're going to lose their work, because they will not. It will save automatically, and the next time they log on, it will be there waiting for them. Once a student has uploaded everything, they just click the data tab a second time, it minimizes, and there's the page as the student left it. Now, at the bottom of every procedure page, a student is going to encounter a series of open-ended questions. And these questions are specifically designed for the student to be able to relate their experimental outcomes and explain what they mean in reference to the concepts being addressed in the laboratory. These are always short answer questions. We do provide the instructor a detailed answer key to ease the grading effort on this. I've yet to find a technology that can sufficiently auto-grade these open-ended questions. When it does become available, we will certainly integrate it into our platform. So those questions can be auto-graded just like the interactive multiple choice and drag and drop questions already are auto-graded on the platform. Now, for time's sake, I'm not going to go into the second exercise, but it's a traditional blood typing exercise. I just want to move ahead for, again, time's sake, to the exam section labeled evaluation. Now, once a student gets to this point, they are provided again with time allocation, a summary of all the learning objectives from the prior 2 sections, so the student can reflect and be assured that they have accomplished all the goals set forth for this lab. And then finally, the student begins the evaluation. As soon as they do, they get a notification that the lesson is now locked, meaning the student cannot page back to look up answers. Once the student starts the evaluation, all they can do is take the evaluation. You'll notice there is a timer in the upper right. The student is made aware of how much time they're spending on the evaluation section as a whole. Now, it will not time out on the student, meaning if it takes a student more than 30 minutes, it'll just keep going, and the student can take more than 30 minutes. You know, for ADA purposes, we could not put time restrictions on our labs, nor should we. But you, the instructor, can view how much time your students spent completing the section. These questions are always multiple choice in format. Student clicks an answer, it automatically stores their selection, and then there's usually approximately 10 to 12. This particular lab has 11 questions. All these questions, by the way, are tracked to the learning objectives for the lab, meaning that these questions span the whole breadth of the lab experience, from the exploration, i.e. background content, to the step-by-step procedures, to the experimental results, and then to those open-ended questions. So this is that you can think of as a one lab practical that occurs at the end of each lab manual chapter. Once the student hits finish, this is auto-graded for them, and they are immediately provided with a report. Now, if I had been a real student and had been answering these questions, I would see my answers in conjunction with the correct answers here. And additionally, what I would see is any answers that I got incorrect, the system would tell me which page, whether it was in the exploration or experimentation, that information, that content was originally presented in this lab. The last thing I get to do as a student is answer one or more extension questions after the multiple choice exam. And extension questions are designed for students to apply the knowledge they gained from conducting the lab to new problems, new scenarios. You know, in A&P, sometimes we may have medical cases for students to apply their knowledge to. But at any rate, these are always application-based in nature. And again, they're always short answer in format. So that includes the student, it concludes the student experience for going through a lab. We do give the student a toolbar drop down, I'd want to point out before we leave the student view. In the upper left corner, not only does that let them access all their lessons, like how we started this session, it also allows the students to review all the materials required for every lab in their lab manual. This is great for students to use even before their lab kit arrives, but they can review the student supplied items for the entire teaching term. So that way, they don't have to every week or 2 times a week, if they realize they don't have something on hand, have to go out and procure it. They have plenty of notice of all student supplied items for the entire teaching term. Also, we provide students with supplemental documents from time to time. For example, how to assemble a microscope. Now, we saw a video on how to use a microscope, but there's actually a supplemental document on how to assemble one if they purchased one from us. And then also, if the student's kit contains dissection specimens, there's detailed instructions for how to handle and dispose of said specimens. And then lastly, for students using the virtual microscope version of the kit, this is where the tool for the virtual microscope resides. And like I said, if anyone wants to see that at the end of the session today, I'll happily give you a very quick demonstration of the V-scope. But what we're going to do now is we're going to shift gears totally. And we're going to see what is designed for instructors on the cloud-based lab manual. Meaning, how does an instructor view the lab manual? How do they view their students' progress? How do they grade those open-ended questions for students? And what tools have we made available to let you customize our existing lab manuals? Before we do that, I'm going to take a quick breath. I will ask the panelists if any question has come through that you'd like me to address that regards to students. I can do that before we talk now about instructors and instructor tools.
Thank you, Dr. D. We had tons of great questions come through, and I want to toss a few. There's about 3 topics that these questions are clumped in. The first one's going to be pretty easy to answer. So a professor, when they logged in and you were showing the lessons that are included in this specific unique course, they had said, am I stuck with these lessons? I like to teach histology, but I don't like to teach what you have included here. Can you talk briefly about how those lessons in this specific course are chosen? And that's where the customization potential comes in, where we have this pre-built experiment library of content that the instructor can pick from and build their course from. So can you talk a little more about that?
Absolutely, Audra, and thank you for the question. So what we've put together for these demo purposes, and they're multidisciplinary, by the way, although today is certainly for anatomy and physiology, is I've just taken a subsample of our library, a very small 5-lab subsample for each discipline that kind of spreads the breadth of our library for that discipline. So when we were here, I'll go back to the lesson list. Now we're in instructor view, but anyway, here I tried to select things that would topics that were pretty universal, but a mix of first semester and second semester A&P. So meaning, yeah, I know that in most A&P courses, we don't cover the blood or the digestive system or urinalysis until a second semester, whereas in the first semester, we tend to focus on histology and cells. We tend to focus on the muscular and nervous systems. So I've got a couple labs that would be a typical first semester example here. But my A&P library consists of over 30 unique labs. So I want you to feel comfortable and confident that we address all important topics that are covered in standard A&P courses, laboratory courses. And as an instructor, you have the ability to pick and choose in that library and choose the labs that you want to use in your course.
Thank you, Dr. D. A couple other topics here. The first one that I want to toss out at you is this idea of cheating. So we had a couple professors say, listen, I teach multiple courses in a semester. They could be the same exact topic. You know, I could be teaching 3 A&P courses at the same time. I worry that my students are going to cheat. Do the evaluation questions change? How do we prevent students from quote unquote chegging is what the term is, you know, cheating pretty much? Do we have anything and processes set in place to diversify these questions to prevent students from getting creative and cheating?
Well, you know, cheating is an age-old problem. It is not unique to online education by any stretch. The methodology in online education may be a little different than cheating methodologies by students in face-to-face environments, but it's problematic in both worlds. I would point out, and we haven't talked about this, we'll see examples here in just a moment regarding learning management system integration of our lab manual into your learning environment. Certainly with that level of capabilities, I would state that any type of online proctoring that you offer for students taking exams in your learning management system, that would certainly carry over into our digital lab manual and the questions that are present in it, since it would live then in your learning environment. You know, as far as changing up questions or as far as what students can access online, we've spent countless hours tracking down various online reporting entities and the answers that they sell to students or anyone who wants to buy them. in what we found and what we've collaborated with similar businesses that have tried to put a stop to this is that this is a very prolific dark world on the internet, if you will. And as soon as we shut down one, another one pops up. It's never ending. And if I change questions tomorrow, within 2 weeks, those questions are already out there. So not to sound like a defeatist, but the reality is that that's how things work in the dark web today. So yeah, could we go in and change questions every week? We could, but then the way your students get pushed that information, it doesn't work from an IT standpoint. So the best we could do would be to run to stand still, and I'm not even sure we can do that. So I would encourage all schools on their end to whatever anti-cheating devices they have installed in their learning management systems. Our lab manual will fully integrate into that, and you should certainly take advantage of those tools.
Thank you, Dr. D. I wish there was a clean solution to this, but students they are clever. Thank you for speaking in more depth about it. The last
I hate to cut you short, but I'm looking at my clock right now and knowing how much time we have for this session. So I think I need to get started on the instructor stuff. And again, I'll try to wrap up early enough to answer questions at that time. But for folks that don't have questions, and that they have a hard stop at the end of the hour, I want to make sure they get to see everything, if that's okay.
We'll hold it to the end. Thank you, Dr. D, for being time conscious.
Okay, no problem. So this is instructor view, and I'm not going to go as painstakingly through the lesson as I already have. We've already seen one. What I want to do is show you what's unique about instructor view versus student view. One unique thing is you can include notes on the landing page for your students. We saw that note earlier, and we see it again here. As an instructor now, I can go in and delete that note in real time for my students, and I can add a new note as I see fit. But more importantly than that, I can really customize my course as an instructor. One way that I can customize it is I can change the ordering of the lessons to fit the flow of my lecture. I can also determine if I want students to complete lessons in a sequence, meaning in this instance, a student would have to complete histology before beginning the justice system, or if I want to open it up and let the student go ahead and begin digestive system before completing histology. So anyway, I just click and unclick the check mark here in order to do that. So let's see what tools I have as an instructor within a given lesson. First of all, the view of the questions and all is the same that we saw for the student. So if I had live students and this was a real course as an instructor, I would see all the answers to these questions up front. But due to demo courses and them having to get repeated time and time again, they work a slight bit different. But what I do want to point out here is that on every content page, so every page of the exploration that there's actual information, then there are a series of these flashing blue bars in instructor view. And every time you encounter one of these blue bars as instructor, you have an opportunity to add supplemental content to the page. That supplemental content can exist in the form of text. It can exist in the form of videos or images. So you can drag and drop supplemental images. You can drag and drop MP4 files, which is the format for videos that the platform will accept. So if you already have a collection of laboratory content that you would like to intersperse into our existing labs, this is a great way for you to accomplish that. And you're not limited by the amount of information you can upload. Any information that you upload, you notice it appears in this slightly gray tint. In the student view, it's much more of a bolder, beigeish background, and it really calls the students' attention to it compared to the standard content of the lab. So the students automatically focus on any notes and supplemental content that you have included for them. Now, you're not limited to just doing this in the exploration section. You also have an opportunity to do this within the procedures. So let's say you taught this for a semester, and your students, maybe they had a question for you on a certain step, although I would certainly hope not, because we should have picked up on that already. Or maybe you wanted to supply a special note for your students somewhere. Again, you are going to have these flashing blue bars in this section too. Maybe based on how you teach this particular topic in the face-to-face environment, you've filmed something about your class, and you can upload that video of those procedures or those outcomes even right here and make it live on the lab manual page for your students. So it's a great tool, again, for you to be able to customize the existing content of any of our labs to best meet the needs of your course. Other tools that you have as an instructor is that you can upload additional supplemental files for your students. So you'll see the same two that we saw earlier in the student view, but you can upload additional files. So if you had Word documents, PDFs, or other types of text and image files that you wanted to share with your students, and them to be able to download onto their computers or print or whatever, you can use this as a repository for those documents. We provide you with PDF answer guides if you want them in print. However, the gradebook tool is much more efficient at allowing you to grade students in this environment. One thing you can do is take a quick snapshot of where your students are at from a grading standpoint and a work submission standpoint in your course. Now, you'll notice this instructor's course has some rather noteworthy students. Looks like, you know, we're never too late to learn a new trade in life, like we see several of these, you know, society stars. But more importantly, you'll see that everyone in this class has submitted 4 labs. And that 3 individuals in the class, the middle 3, have had grades assigned to those labs. So as an instructor, this means to me is that Snoop and Betty have not been graded yet, and I need to go in and assign them a grade, and then their circles will be filled in. How do I assign grades? We're limited on time, so I'm not going to dive too much into this, but I assign grades in each lesson for my students by assigning the weight to every single question. So all of those drag and drop questions, multiple choice questions, data tables, image panels, etc., have a point value associated with them. Once an instructor sets up the point values and weightings for each of these questions, they're stored in the system. So every semester that you teach, it carries over automatically, unless you want to go in and change them semester to semester. To grade a student, and again, we're time crunched, so I'm not going to spend too much time on this, but you would simply click on one of their labs that they have started on and submitted, go into the exercise, you will see the answer key image, and then directly below it, if this student has uploaded a photo like they were instructed to do, you would see their photo immediately below it. Then you can assign a point value for the student's submission and leave them a note of why they got the score they got. The student, in their view, also has a grade book, but the only thing they see in their grade book is all the comments you left for them and the grades you gave them for each submission. So this is a great way in real time you can communicate grades to your students and they always have a view of them. So a great communication tool. You know, you're not having to manually mark up a lab report or anything like that. It's all digitized here within the lab manual itself. So let's talk about a couple of other tools that you have available. And probably the most exciting one is something we call custom lesson authoring. And again, for time's sake, we're not going to build a lesson. But I want you to know that we've designed within our digital lab manual the opportunity for you to create any of the elements that you've seen today on my shared screen into a unique lab for your students. So if you wanted to create, let's say, an extensive background content, i.e. a lecture for your course, and you wanted it to look like just like the lab background sections, you can go in and create just a lecture for your students and upload all the content you want, all the images. You can build all the questions you want, totally free for you to do. Similarly, if you wanted to go in and create an exam for your student, a comprehensive exam over the whole semester, so a true lab practical, you can go in and just create one big exam and put it at the end of the semester, and the student has as many questions as you choose to build for them. And then lastly, you know, if you wanted to create maybe a research project for your students or some type of pen and paper type exercise for the students and truly format it just like you've seen our labs formatted, you can create a 3-section lab, you can create activities, all of that for your students. We give you all the training and tools you need to be successful in creating content just like our internal team of curriculum designers have access to. And then lastly, we give you all of the data you need to allow your IT team to fully integrate our lab manual within your learning management system. So on the most basic levels, you can simply direct your students to our cloud environment by copying and pasting an enrollment link. Or again, from an integration level, we offer a variety of links and course hooks and individual lesson hooks for every lab in the lab manual. So you can move around all the topics and place them adjacent within your learning management environment to your lecture. So again, it's a seamless experience then for the students. Any type of, again, grading tools, proctoring tools, submission tools, anything like that you already have existing in your learning management system, you know, now they can be applied again to everything that you have linked. So, so truly seamless for students if that's what you choose to do. And again, we do have a variety of resources to assist you and your IT team with that level of integration. So that includes the demo portion of today. Like I said, I want to get finished in time to answer any further questions that you have. You know, if there's enough demand for looking at the vScope, we'll do that. If it's not a popular request today, then we don't have to. That's fine. So I'll turn this back over to our host and answer any questions that need to be addressed.
Thank you, Dr. D. And thank you, everyone, for all of the great questions and the engagement. We have a couple questions for you. We've received multiple inquiries regarding the student's safety and liability, a lot of which were geared towards the student's handling of the urine and blood samples. Could you go into a bit further detail for us today, Dr. D?
Well, students are only handling their own urine and blood samples, first of all. So they're not coming into contact with any foreign materials there. It's standard laboratory protocol for how students are taking blood in that they're, you know, disinfecting the surface before they use a sterile single-use lancet, and then again disinfecting it the second time after, the blood draw of 1 to 3 drops. So very minimal side of the fingertip puncture on that. In the urinalysis lab, and we provide sterile urine cups and wipes and all of that in conjunction with that activity. But again, all of our kits, keep in mind, are designed for single student use in a remote environment. So this is not a traditional lab environment where students are coming into contact with other students' fluids in any form or fashion.
All right, thank you. What on the same token, what happens if a student has an accident at home? Are they insured? Have accidents happened at home in the past?
So we're first and foremost, yes, we are the liability provider for your students using our kits and our curriculum. Can students have access? I suppose. We have never had a claim against our insurance in our 20-year history. And I think that's not because students are not accident prone. I think it is more of that the materials that we provide students are very safe, either safe from the type of material that we provide the student with, or safe from the volume of chemical that we provide the student with, and certainly safe from the standpoint of how we dictate the student use the materials that they've been shipped. And I know if you haven't taught online before, and I did see that poll that came in earlier, and it seems like 63 or some odd % of the respondees had not taught lab science online. I know that the train of thought is strictly with our experiences of teaching on campus. And I have to reiterate that, again, the materials that students are receiving and the types of procedures that they're conducting, while they are designed to mirror the overall campus experience by the results that they produce, they're not mirroring necessarily the on-campus experience from exactly what the student is using and how they're using the materials. So everything is really been pared down. I mean, the only things I can't pare down for your students are the dissection specimens. So if you want your students to dissect a sheep brain, a sheep brain is a sheep brain. And same thing for a sheep heart or a fetal pig, So aside from the actual specimens for dissection, all other materials in your students' possession when they're conducting these experiments are significantly smaller by volume than what your on-campus students are doing. So again, as long as the student proceeds as directed, there should be really a minuscule chance of them harming themselves in any way. And we stand behind that. So, and I understand the concerns from a safety standpoint of each instructor and institution, but we stand behind our lab procedures and materials.
Great, thank you, Dr. D. And then because we are running short on time, if we could get a demo of the vScope, we've had a lot of interest in that and a lot of requests.
No, well, we usually do, so I would have been more surprised if we didn't see it. Okay, so what is the vScope? vScope stands for virtual microscope. Our software engineers developed this for us. It is our property. It is not third party that we're borrowing or paying royalties on or anything like that. The important part of it being ours is that the slide library is identical to the physical slides that students using a real microscope would receive in their kits. And indeed, that's how we developed the slide library, by using our physical slides. Students are required to manipulate this microscope exactly like they would if it were real. We provide that prerequisite lab that we talked about early on, and then we've got some cheat sheet steps for the students should they have forgotten what was covered in the prerequisite lab, to make sure they realize the proper sequences of focusing and using slides and capturing those images. For example, we have to lower the stage on a real microscope in order to move slides on and off safely. We need to do that on the virtual microscope also. We obviously can't view a slide until we turn on an illuminator. Then we should always have the lowest power objective selected before we place a slide on the stage. Once I've done that, as a student, I've got 3 green checks. Now I can select a slide from the library. The titles of all the slides in the library are identical to the titles of the slides that are called out in the lab manual. So if I click on the first title here, you'll notice as a student, I see this horrible blurred image, and it's not even a complete image because I'm not even in the middle of the slide. Not unlike if I had just put a slide on a real microscope. So I need to both center the slide, and I need to, and I do have 2 centering tools, just like a real microscope. And then I also need to adjust the coarse and fine focus knobs in order to properly view the image. Again, not unlike a real microscope. These are all keyboard controlled. They are ADA compliant for that reason. So it means students do not need to use a touchscreen, nor do they need to use a mouse. They can use the up and down arrows on their keyboard should they need to. And again, we give students a keyboard accessibility cheat sheet should they want to know how to do that. We also give students a cheat sheet for microscope controls. Should they forget what the difference between a coarse and fine focus knob is, or between an objective and an ocular lens. Once a student is focused on an image, they can then change the objective powers. They can repan around the image to select a particular cluster of cells in this standpoint. And once they are happy with the view that they have achieved, they can download that image. Now, if you recall, in the lab manual, students are required to include photographic evidence that they have accomplished the procedures. And if we are talking about histology, or not unlike when we saw in that blood lab, students have to upload images of their focused slides. So you notice I click download image. Here is JPEG of this image. I can now drag and drop that JPEG file into the image panel of my lab manual. It's as straightforward as that. Just as if I had used my cell phone camera through the ocular lens or through the barrel of my microscope to make a real microscope photo, then once I have that image on my phone, I can again transfer that into the image panel within the lab manual. So very straightforward, whether we're using a real microscope or in this case, the V-scope. But I hope you see with the V-scope that it is not unlike a real microscope in that students do have to manipulate the device in order to produce the desired outcome. This is not merely a collection of slide images that a student's just downloading. If a student doesn't focus the image properly, if they don't find the right features on the microscope slide by panning right and left and up and down, and they download the wrong image, then they download the wrong image. And so, again, they're not necessarily at any advantage compared to students using a real microscope. So, if you have any quick questions, I know we're already out of time. If you have any quick questions about the V-scope, I'll happily address those.
We had one. Can labeling be accomplished with the V-scope?
Labeling is accomplished in the panels that students, so let's go to student view. So these image upload panels for students, we'll click one here. So as soon as I move my image into here as a student, as I drop my JPEG file or PNG file, any type of image file will work, I get a little toolbar that pops up, and I get a view of a garbage can, and I get a view of an annotator, and I use the annotation tool then to label my image. So I can type in labels and drag and drop them anywhere around the margins or on particular features in the image. So very important. We didn't cover this in the demo because we're always time limited, and there's far more features within our Lab Manual platform than we ever have time to get through. But we assure that any software that a student needs to be successful with submitting their work is incorporated into the digital lab manual functions. So meaning labeling tools, annotation, drawing tools, all those types of things. If we're asking a student to label an image, we're going to make sure that the panel that they upload their image in has a labeling tool turned on that panel. So then the student uses that technology to label. Your students are not expected to have access to Microsoft Office, although I know most of them do, meaning they don't need to have a Word program or a PowerPoint program to label something. All the functions they need are embedded within our digital lab manual.
Thank you so much, Dr. D. All right, so for the questions that we were unable to address during this presentation, your dedicated distance learning specialist will reach out to you directly with clarification on those questions for you. We apologize again, as we are limited by time. We do apologize, but we want to thank you for your time and your engagement today. And we just want to remind you to keep an eye out for the follow-up email. It will have the recording of the webinar and any other resources that we shared during this presentation to help aid you in your transition to online. So thank you so much again. And we really appreciate the participation and the engagement, and you guys take care.